I am absolutely delighted to welcome Masaru Nobu from the National Institute of Advanced Industrial Science and Technology from Japan, who makes us a great pleasure of not only joining us and giving a talk, but also doing it in the middle of the night for him. So again, thanks a lot for making this extra effort on top of everything else. So his work has been focused on really characterizing novel microbial metabolisms, especially connecting genome metabolism and physiology. And one especially exciting aspect of his work has been the ability to, and recently, cultivating some organisms that had been only known from metagenome on data so far. And that led to really new insights into methodology, for instance, as well as the identification of unique cell membrane structures in several new lineages. So with all that, just reminding you quickly to please put your question in the Q&A option and kind of chat thing on the right side of your screen. And without further ado, I will hand it over to Masaru.
Okay, thank you for the introduction, Simon. As introduced, today I'd like to share our research on an archaeon at the prokaryote-eukaryote interface. As you all very well know, there are three domains of life on Earth: bacteria, archaea, and eukaryotes. So eukaryotes are quite different, as you all know, because of their cell structure compared to archaea and bacteria. And thus far, it has been thought that eukaryotes and archaea are closely related, but their exact relationship had remained unknown, primarily due to the low diversity of archaea that we have been able to culture and characterize. So what does that mean? That means that we are not quite clear yet on what kind of transition happened from simple cells like archaea and bacteria to complex cells like eukaryotes. So current theories. It's pretty well accepted that archaea and archaeon and bacterium fuse together to form eukaryotes. And although still debated, it is thought that a group of archaea called Asgard-archaea that was discovered through metagenomics was likely the host archaeon. And what is even more hotly debated is whether the host archaeon was already complex, already on its way towards the cell structure of eukaryotes, or not complex yet. And so what we want to know is what kind of structure the host archaeon may have had, and also what kind of properties the archaeon had that set them apart from other archaea. In other words, what set them up for the cross-domain fusion? And why did it have this potential to begin with? And in this talk, I'd like to talk a little bit about what we want to know. So what we did is we went out to sample deep sea sediments where there's tons of uncultivated archaea in 2006. And our goal was to culture these unknown archaea to really uncover, better understand the diversity of archaea, what they're capable of, who they are. And so we took the sediments, took it back to the lab, and we cultured it in a down-hanging sponge reactor. The reason we used a reactor initially instead of directly going to agar plates or serum bottles was we wanted to acclimate these organisms to artificial media and also the higher energy conditions that we grow them under in the lab, because we really need these organisms to grow to characterize them, which they're growing probably very slowly in situ. So we do this kind of enrichment over years, and then we take this pre-enriched community and further enrich them in serum bottle cultivation, providing different kinds of substrates. And through over 10 years of cultivation, we have been able to isolate a novel archaeon that we named Promethearchaeum centrifugum strain MKD1 using casamino acids and formula milk. So we sequence the genome and characterize the culture through experiments. And based on the genome, we construct a ribosomal protein tree to look at the relationship between this archaeon with other archaea, bacteria, and eukaryotes. And we find that it is the first archaeon that bridges the gap between archaea and eukaryotes. So this represents the first cultured organism that allows us to construct a tree where eukaryotes branch from within archaea, showing that the third domain of life likely originated from the second domain, from within the second domain. When we look under the microscope, we found few surprises. So the organisms, the MKD1, is very small cocci, about 550 nanometers in diameter. And looking at the cells, we find no visible organelle-like inclusions. In other words, although the ancestor of eukaryotes was expected to be an archaeon had been proposed to be an archaeon with complex cell structures, this strain does not have this. And of course, this strain does not represent our ancestor. It is another descendant of an ancestor that we share with Archaea. But given its structure, it's likely that our ancestor Archaeon didn't have organelles yet. Although the inside of the cell was pretty simple, we found that the outside of the cell was relatively complex. Protrusions have been found in many archaean bacteria, but this archaeon has a unique type of protrusion reaching out of the cell, and these protrusions can reach up to 20 to 30 times the cell diameter, which is quite interesting. And furthermore, you can see here that some of these structures can be branching. Another feature of this organism is that it's very, very, very slow at growing. It's very bad at growing, at least from our perspective. Its doubling time is 14 to 25 days, and so it takes about 4 months for the culture to fully grow. And even after waiting for 4 months, it only reaches a max cell density of 10 to the 5th cells per mil, which is the same cell density as tap water. And to further contextualize this, both the doubling time and cell density is more than 1,000-fold lower than E. coli. And further experiments, microscopy, and genomics shows that this strain MKD1 can degrade amino acids through syntrophic growth, so metabolic symbiosis with other microbes via interspecies hydrogen and formate transfer. And we were able to demonstrate this with both a partner archaeon and a partner bacterium. And we also confirmed that this strain utilizes amino acids using stable isotope probing with nanosims. So this organism that represents another descendant of our very close archaeal ancestor has this kind of metabolism where it degrades amino acids and primarily gains energy through oxidizing 2-oxo acids, which is the metabolic intermediate produced from amino acid degradation. And downstream, it produces fatty acids, hydrogen formate, which can be transferred to partner organisms, and in our culture's case, to a methanogenic archaea or a sulfate-reducing bacterium. So what we want to know now is we want to compare this archaeon's metabolism with other uncultured relatives of this archaeal group, so Asgard archaea, to reconstruct the metabolism, the physiology of our ancestor. And so before we dive into reconstruction of its metabolism, I'd like to mention what kind of transitions need to happen from archaea to eukaryotes. So archaea have no intracellular structures. According to our analyses, this is also likely true for our ancestor archaeon, and no nuclear membrane. And no endosymbiont so far, and physiology-wise, anaerobic syntrophy, and symbiosis dependent. This is all quite contrasting to the eukaryotes, which have an endomemory system, nuclear membrane, mitochondrion, and dependent on aerobic organotrophy, and generally independent. So what kind of metabolism was our ancestor capable of? We compare the amino acid degradation of MKD1 and other Asgard archaea and find that it is quite widespread across all of these lineages. And as some of you probably know, the amino acid degradation pathways, many of them are reversible. So organisms use the same pathway for degradation and biosynthesis. So if we exclude those and look at these blue ones, which are exclusively used for degradation, we see that they are also quite widespread, and at least for serine, methionine, threonine, and histidine, we detect them in at least 4 out of the 5 lineages that are known for Asgard-Archaea. And the same is true for electron metabolism. So it seems like these organisms can all produce hydrogen and also live fermentatively. So it can produce fatty acids through anaerobic metabolism. And through, although I won't show the details, through phylogenetic analyses, we find that the Asgard-Archaea ancestor was capable of degrading serine, threonine, and histidine, and also fermentatively producing butyrate and propionate. So clearly, the ancestor of Asgard-Archaea and also our ancestor had metabolisms very similar to the strain we have isolated. Another interesting feature of these archaea, when we were growing these archaea, because of their slow growth, we thought maybe they aren't so good at making the building blocks of their cells. And we analyzed the genome and find that that is quite true. So here is a table of 22 amino acids that bacteria and archaea utilize. And when we look at the biosynthesis, these dark blue squares represent ones that the organism can synthesize de novo. And the light blue ones require some sort of intermediate precursor to be provided. And the white ones, there's no biosynthetic capacity there. And as you can see, compared to the sulfate-reducing bacterium and methanogenic archaeon that are partners, can synthesize most of these themselves, but all Asgard-Archaea have very, very poor biosynthetic capacities. And this is true for vitamins and even nucleotides as well. So that means they can't synthesize their own DNA or RNA without precursors being provided. So based on this, our ancestor Archaeon lived oxygen-free based on the amino acid metabolism, fermentative metabolism we found, and was likely quite dependent on 2 types of symbiosis. So one, amino acid degradation often requires a metabolic symbiosis, and in addition, if these features that we see in extant Asgard-Archaea was conserved in our ancestor, they needed some sort of partner to synthesize building blocks for them to reproduce. Now, the biggest differences between this ancestor and us is that this ancestor is dependent on anaerobiosis, whereas we, for the most part, are dependent on oxygen respiration. Another big difference was the cell structure. Assuming our ancestor shares the same cell structure as MKD1, this is quite a transition. When we look at the genome, as has been shown for other Asgard-Archaea, MKD1 does have genes that have been characterized, that have been known to be eukaryote specific until the discovery of these Asgard-Archaea. So it is not a lack of these genes that makes this archaeon have simple structure. Even though this organism has simple structure, it still has these eukaryote-specific genes. So like actin, ubiquitin, escort systems, maybe they originally had a different function in archaea prior to the rise of complex structure. But this still requires a lot more research. So in the transition from archaea to us eukaryotes, which was the first hurdle that archaea overcame? Was it O2 respiration or a complex structure? Based on our understanding of prokaryotes and their energy metabolism, we believe this transition from anaerobiosis to oxygen respiration was a huge hurdle, especially because oxygen was absent on Earth when our ancestor existed. So we base our further analysis on how archaea transitioned towards eukaryotes based on the archaea probably depending on the transitioning from anaerobic syntrophy to aerobic organotrophy to start its path towards eukaryotes. So starting from an ancestor archaeon like this, based on our analyses and also other people's papers, our ancestor archaea likely existed on Earth around the time when the Great Oxidation event happened. So when oxygen was first introduced on Earth and started accumulating. And as mentioned earlier, because these organisms are highly dependent on anaerobic metabolism, oxygen was toxic to them. And what's quite interesting is based on our results, MKD1 is very, very slow growing. And if this was true for our ancestor as well, it would have been very difficult for them to adapt to aerobic conditions because of their slow growth rate. And that means because they're slow growing, that means they only acquire genes or new abilities very slowly. So it is possible that rather than evolving to utilize or detoxify oxygen themselves, they relied on organisms around them that are faster growing, faster adapting to utilize oxygen. So in this case, perhaps there is a symbiosis with oxygen-respiring ancestor of mitochondria to detoxify oxygen and also provide amino acids and vitamins. And because after the rise of photosynthesis, the oxygen levels in Earth gradually rose, it was probably insufficient to simply be adjacent with an oxygen-consuming partner and required a more intimate interaction. And this is when the archaeon may have interacted with the mitochondrial partner through using its protrusions and vesicles. And through a process that has been proposed in 2014, where protrusions was a very important component, it is possible, physically, topologically possible for these protrusions to mediate interaction, engulfment of a mitochondrial partner, and actually simultaneous formation of a simple nuclear membrane. I won't describe this part in detail, but what's very interesting is that based on our observations of the number of protrusions per surface area of the archaeon, if these pores where the protrusions were, if these became nuclear pores, again, this is very speculative, if it did, the number of pores per micrometer squared would be about 14, which is on the same order, very similar to nuclear pore density in yeast. Okay, so assuming that that kind of transition did happen, we have this kind of weird transitional state where we have two organisms sharing a single cell. And at this point, through such symbiosis, you could transition from a state where you have no nuclear membrane to having a nuclear membrane-like structure, and also to having a symbiont on its way to becoming something like a mitochondrion. Now, this intermediate transitional state has problems because as in this state, the organisms cannot mature to become a single organism because there are some redundant metabolisms. First, both the archaeon and bacterium were likely synthesizing lipids themselves because they had those abilities to begin with. And they both catabolize 2-oxo-acids. This 2-oxo-acid, including pyruvate, is the primary energy source inside of the cell that both utilize. But it is difficult to overcome this because the presence of this 2-oxo-acid metabolism is in the way. And this is because it is completely dependent on anaerobic conditions to gain energy. And without throwing away this metabolism, these organisms cannot mature as a single organism. And so let's first look at the lipid biosynthesis issue. So this comes back to drawing back to the data that we show that MKD1 is slow growing. There's a huge difference in growth rate between MKD1 and, let's say, rickettsia, which is closely related to mitochondria. And if in this transitional state, both organisms had such growth rates, you'd see that the archaeon would not grow as fast as the endosymbiotic bacterium. And so the bacterium inside would grow faster and also die faster. And so this is quite interesting, where there'd be a release of massive amounts of DNA and lipids, cellular building blocks into the cell. And while this would resolve some issues in terms of the archaeol's ability in synthesizing its own building blocks, that also means there's a lot of organic molecules that it had to deal with that was being released, especially because lipids are hard to degrade under anaerobic conditions. And it is likely that the host archaeon integrated the symbiont's genes and lipids into its own cell, which allowed for the transition of the archaeal membrane to a bacterial membrane, which we see in extant eukaryotes today. And also has been shown by many papers that a huge portion of our genome is bacterial rather than archaeal, and likely this kind of process contributed to that. And in 2018, it was shown that the archaeal and bacterial lipids can mix in the same cell. So this kind of transition can indeed happen. Next, the catabolism. This is a major hurdle because one of them needs to stop doing 2-oxoacid metabolism, but stopping 2-oxoacid metabolism means forfeiting gaining energy itself. So how do we get past this hurdle? We believe that the trigger for this was the symbiont becoming parasitic. So why is this? So typically, the archaeon and bacterium, they were gaining energy themselves. They were absorbing metabolites, organic compounds from the outside and generating ATP themselves inside the cell. But once the bacterium is inside the archaeal cell, it's put under very unique conditions for the first time in the life's history, where there's raw energy in the form of ATP available around itself. And it would make a lot of sense for a mitochondrial ancestor to absorb this ATP as raw energy. And indeed, the mitochondria have developed ATP carrier protein, which transports ATP across the membrane, and this protein is only found in mitochondria. So the motivation for evolving this gene was unique to its evolutionary history. So when this happens, the archaeal host needs to deal with this somehow. And one of the few ways that the archaeal host can deal with this is to actually stop its own metabolism and stop ATP synthesis on the archaeal side. And what this does is, because this protein AAC has been demonstrated to be reversible in previous work, by stopping ATP production from the archaeon itself, it can actually reverse the flow of ATP from the mitochondrion towards the host archaeon. And this allows to kill 4 birds with 1 stone. So through this process, the host can lose catabolism and reverse the AAC activity, so reverse the parasiticism, and also tame the bacterium as a symbiont that provides ATP. And this also resolves the functional redundancy and also allows it to overcome the oxygen sensitivity. So through such a process, we have the mitochondrion forming as an oxygen respiring and ATP providing symbiont, and also transitioning completely from anaerobic to aerobic degradation and symbiosis to an independent state by having a symbiont inside of the cell. And through such transitions based on the observations that we had from the strain MKD1 in comparative genomics with other Asgard archaea, we have constructed a new model that I just described called the E3 model, in which the aerobic bacterium became the engine of the cell, and the archaeon became the pilot of the cell. And through this process, because the ATP production, energy production, is completely delegated to the cell-bound bacterium, the host archaeon is released from energy metabolism. And I won't describe it in detail here, but energy metabolism has a lot of physical limitations, as in there's a size limitation. So that is likely why bacteria and archaea are small, whereas eukaryotes are large. But because this archaeon was released from this restriction, allowed it to start exploring larger sizes or different cell structures. So in total, based on the culture of MKD1, the first Asgard archaeon, we were able to show that it has slow growth, oxygen sensitivity, symbiosis, and unique cell structures, which can all be linked to potentially helpful features for generating the first eukaryotic cell. And what we need to study next is this organism, MKD1, has a very, very large genome, about 4 megabases. And this is very large for its cell structure, cell size, and also for its slow growth. Why does it have such a large genome? And also, why it produces such protrusions is still unknown, and how it uses the eukaryote-like genes still remains unknown. So once we can link these to the features that we've described and related to the model above, I believe we can get to a much more accurate model and understand much better how we evolved from archaea. And so with that, I'd like to conclude my presentation on our research that helps us understand the path from archaea and bacteria to eukaryotes. And I'd like to thank my collaborators. This project would have certainly not been possible without all of their help, especially Hiro Imachi, who is a specialist in cultivation. And with that, I'd like to take any questions you may have. Thank you.
Thank you so much. It was I'm doing the round of applause here, but this was fascinating. And that's perfect timing too, because we do have time to take some questions. Let me start here from Taiseo, and I apologize if I'm not pronouncing the name right. Kind of summarizing, but I think the question is also relevant to really relevant to what you just described. What are in your mind the ways that you would be able to minimize the gap between the understanding we have from physiology and phenotypes in the lab and the real metabolism, physiology, and phenotypes in the native habitat, which is more than challenging to replicate in this case?
So because this organism is quite slow growing, this is very puzzling. So we are on our way to try and understand how this archaeon actually lives in situ. One of the big gaps that remains is that because we want this archaea to grow as much as it can, we throw a lot of energy at it. We provide relatively high concentration amino acids and formula milk for it to grow, but it's not even putting a dent into it. It's using less than 1% of what we provide to grow in our bottles. And no matter how long we leave it, 99% of what we provide is not touched. And the archaeon stops growing after 4 months. So at this point, we're trying to figure out why that is happening. Once we can understand why that is happening, we can start to better understand what the actual conditions it prefers for growing is. But it'll probably take quite a bit of time, given that it takes about 4 months to grow this organism.
That makes sense. Thank you. And the second question from Roland, which actually, I guess, a two-part question. The first one being more like this comments on your question on why so many genes and why such large genomes. And it's true that, as Roland say, some genes could be present and simply not expressed or very rarely expressed. But then his suggestion and his question about have you tried this is, to my it's a really great idea, at least to me. Have you tried massively parallelizing this culture in micro droplets? So encapsulating in micro droplets one cell of your archaeon, and then either different conditions or different potential symbionts, and then screening the droplets for any change. In this case, I think he was thinking of change in subcellular organization that could be visible once the archaea has been kind of incubated in these microdroplets. Is this something that you have on your horizon, maybe?
So yes, that it would be fantastic if we could do that. Right now, our main hurdle is getting this archaeon to grow a little faster and to higher densities. So right now we are trying to optimize the medium, so that allows us to explore a lot more different types of experiments, and also culturing other relatives that may grow faster, may grow to denser cultures. Because the reactor that I showed earlier, that Hiro is running right now, that reactor still has many, many, many diverse Asgard archaea in there that we haven't cultured that are belong to different lineages. So I think step by step, we'll try to grow this archaea better and also get potentially better model organisms for this group of archaea, and then explore such interesting experiments.
Sounds good. Looking forward to it.